So good afternoon, everybody. My name is Leyre Fraile, and today I'm going to explain in a very brief manner how we can measure magnetic nanoparticles that are deposited on a rigid and flat surface using a radiofrequency refractometry sensor. First, let me introduce myself. So I studied biotechnology at the University of Oviedo, and then I completed a master's in nanoscience and nanotechnology in Madrid. Now I have just begun my PhD. So now that we know what this work will be about, the next question we need to ask ourselves is why? Why are we interested in measuring solids? Nowadays, nanoparticles on surfaces has helped making advancements in areas such as biosensors, electronics, and optics. Therefore, we thought it would be useful to measure the signal of those nanopatterns. Another potential application, more bio-related, could be the magnetic separation and measurement of cells that have specific receptors attached to the magnetic nanoparticles. Looking at the literature, we can see that scientists have been trying to do this for many years now. So let's think how we could perform this. As we all know, blood is an heterogeneous liquid with many different types of cells. Let's imagine we have a sample with two different types of cells, green cells and orange cells. And we want to separate and measure the concentration of the green cells. So we can add magnetic nanoparticles that have specific receptors so that they will attach selectively to the green cells. And then with a magnet, we can separate those cells that have the magnetic nanoparticles inside of them. Then we can grow those cells in a Petri dish so that if there are any adherent cells, they will bind to the surface of the dish. And by multiple washings, we remove the nanoparticles that didn't go inside the cells. And now we have green cells with magnetic nanoparticles ready to be measured. So now that we have introduced the theme and the motivation behind this story, it's time to reveal our main character. How are we going to measure these magnetic nanoparticles? So let me present you the radiofrequency refractometry sensor that we are developing at our lab. It has resonant coils as detectors so that it can detect and quantify magnetic nanoparticles with a great sensitivity. How does it do it? The method devised for nanoparticle quantification uses a planar coil, tracking its self-resonance frequency as different nanoparticle masses are positioned on top of it. This is how I'm going to represent the sensor from now on. So measuring on solids is not that easy. Let me represent the sensor in green and the sample that we want to measure in blue. In order to make a good measurement, we need to apply an homogeneous pressure system. We also need to accommodate the sample so that it will always stay on the same position on top of the sensor, and we also need to design a sample holder that contains the sample. So in summary, we want to measure rigid samples, but as in every good story, there are always some complications. Therefore, we have divided this work in three parts we need to complete in order to reach our goal. Let's begin with the first part. To ensure repeatability, as I mentioned previously, the sample must be flattened against the sensor surface, maintaining a consistent distance for the nanoparticle deposition in every measurement, and that can be achieved by applying pressure. So we designed the pressure system using a 3D designing program and a 3D printer. The clamping force, which compresses the substrate, affects the refraction index environment, keeping it constant between measurements. Then we designed the sample holder. We created some models of sample holders, each of them designed specifically for the substrate that was going to be fit inside of it. We tried out different materials as a substrate for the sample, attending to the homogeneity of the bulk material, the thickness, porosity, rigidity, and surface properties of the materials. We tried with wood, plastic, alumina, and different types of glass. So now that we have designed some sample holders and we have some rigid substrates to compare between them, we can go to the next part of the project, which is synthesizing, characterizing, making the positions, and measuring the magnetic nanoparticles, using those designs from earlier. We synthesize the magnetic nanoparticles based on manganese ferrites, using the hydrothermal coprecipitation method. This technique involves the precipitation of nanoparticles when a mixture containing iron cations 2+ and 3+ comes into contact with a basic solution. The magnetic nanoparticles were then coated with citric acid, and we obtained a sample of nanoparticles with an average size of 10 nanometers and a round morphology. Now it's time to try them out. So first, we deposited the same volume of magnetic nanoparticles on top of the 3 substrates and obtained these results. As you can see, the clear winner is glass, followed by alumina and plastic. But although the best result was glass, we decided to use alumina instead to perform the calibration because it didn't break easily, and also because the magnetic nanoparticles attached more to its surface. Then we did a calibration of the magnetic nanoparticles. We deposited these magnetic nanoparticles on top of the sample folder using a pipette coupled to a 3D printer. And these are the results for the calibration with alumina, with a pretty good R-square. So now we only have one task left to achieve our goal, and that is the bioapplication of this project. We are going to measure magnetic nanoparticles that are inside cells adhered to a glass as a proof of concept. To do that, we made a cell culture using HeLa cells and a 6-wall plate. In all of them, there was a glass as a substrate, so that the cells would grow attached to it. In the first column, we just grew the cells as a control. In the second column, we incubated the cells with 100 micrograms of magnetic nanoparticles per milliliter. And in the third column, we put twice the number of magnetic nanoparticles per milliliter. And then we left them for 24 hours to incubate. As you can see in the confocal images, there is a clear difference between hour 1 and hour 24 after the incubation. It's clear that the cells were pretty hungry and have successfully eaten the magnetic nanoparticles. Now we did a cell counting using the Neubauer chamber, also called the hemocytometer. And as you can see, we have a pretty good amount of cells. Finally, we measured the cells with the magnetic nanoparticles inside, and these were the results that we obtained. As you can see, we obtained twice the signal from the cells that had the double of magnetic nanoparticles, which is what we expected. So we can conclude with this that we have achieved the goal of measuring on rigid substrates with our sensor. As plans for the future, we would like to analyze and compare more possible substrates that can work with this new system so that we can make better calibration curves to obtain validation parameters, such as the detection limit or the quantification limit. Also, we want to measure 2D arrays of nanoparticles. And finally, we would like to do the functionalization, bioconjugation, and direct targeting of cells using the magnetic nanoparticles as biotags. So with this, I finished my presentation. I would like to thank my group and to you for listening to me. Thank you.
Thank you very much for your talk. Please, any questions? I maybe have a quick question to start. So in your last data plot, you had quite large error bars. So I was wondering, like, do they come from many different samples that you analyzed and then did some statistical analysis?
This one, you mean?
How did you get the statistics from this measurement, from this experiment?
That is because we are still trying different substrates and different ways of adjusting the plates on top of the sensor. So since we are only in the first steps of designing this new measuring system, we still have a lot of margin to improve ourselves and to improve the measurements.
Okay, so the error bar is like an experimental, due to experimental, not different.
Yeah, and also because here the cells were cultured on top of glass, and as I told you before, glass didn't have the best results because it broke really easily. So to obtain a good statistic, we had to repeat the process lots of times, and maybe the repeatability isn't quite right. with alumina, I think we could get better results because they would attach better.
I see. Okay, thank you. Any more questions? If not, then we can thank our speaker again.